Okay? So how do we go from DNA to protein? So we're going to study two different stages that help us accomplish this: transcription and translation. And just to kind of start off this lecture, I think I should introduce you first to what's known as the central dogma of biology. Okay, so the central dogma of biology states that we use DNA to help us make RNA, and we use that RNA to then produce protein. Okay, so this is the central dogma of biology. Now, quick question, actually, before we go into anything as to where transcription and translation play roles in this. Go ahead and help me classify these different types of macromolecules. First off, we'll start with the easiest. What classification of macromolecule is a protein? A protein, good. What about DNA or RNA?
Nucleic acid.
Nucleic acid. The NA stands for nucleic acid. Okay, so think about transcription and translation in the context of language. Okay? Right now, as I'm speaking, all of you are transcribing what I'm saying. Is the message changing languages at all? No, it's staying the same language. It might be shorthand, and it might be slightly different, but hopefully the message and the words that are coming out of my mouth are represented in some degree in English on your paper right now. Okay? So you're transcribing. So if it's between DNA going to RNA or RNA going to protein, where do you think transcription is going to fall in this particular case? DNA to RNA. Good. So this process that takes DNA and converts it into a message in RNA is called transcription. We're not changing languages. Okay? But now if I told you, hey, I want you to take what I am saying and I want you to write it down in German, okay, or after you've written it down, you need to change it to German. Now you're going to translate it. Okay? So when we're going from nucleic acids to protein, now all of a sudden we're going from one classification of a macromolecule to a different classification, and we are changing the language. So this is where we're translating. Okay? So that's translation. Alright, so these two stages govern all of this. Here's the bad news. Transcription is broken up into three substages: initiation, elongation, and termination. Translation is broken up into three substages: initiation, elongation, and termination. Yes, it is annoying, Hirsch's, but I'm going to try and be your spiritual guide through transcription and translation. Okay? So, this first part, I believe, is actually asking the question, how was all of this brought about? I'm probably not going to spend a lot of time on this, and you shouldn't spend a lot of time studying it, because it's just a bunch of older guys who discovered all of this and formulated these hypotheses. Basically, all these first couple of pages are saying is initially scientists had a one gene, one enzyme hypothesis. So every gene in your body gives rise to a single enzyme. Good starting hypothesis, and it was later, I guess, fleshed out or better elucidated to be a single gene, single protein hypothesis. So one enzyme, one gene hypothesis. But some proteins aren't enzymes, so then it was, I guess, reestablished as the one gene, one protein. But then, they were like, well, wait a minute. A lot of proteins are actually quaternary structures composed of multiple polypeptides. So then it became the one gene, one polypeptide hypothesis. Now, what you're going to need to know is that when I learned this in school, I learned all polypeptides as proteins. So if you hear me saying, this is how we make the protein, what I'm actually saying is, this is how we make the polypeptide. Okay? When we're talking about transcription and translation, especially translation, you're probably going to hear me say protein a lot. When I'm saying that, I'm actually meaning the polypeptide that is made during translation. It's one of those things where I've just been taught so long to say it one way that it's really hard for me to remember all the time to say it as polypeptide. Okay, so here's what happens. Yes, was there a question? No? Okay, I'm not going crazy. All right, so we have the DNA inside the nucleus, and we're going to utilize an enzyme called RNA polymerase to make a specific type of RNA off of that DNA message. It's only going to bind in specific regions of the DNA, these locations called genes, and it's going to make this RNA that's going to carry the message that will eventually be translated into protein or polypeptide. Translation occurs at the ribosome, which is going to be in the cytoplasm, and it's going to convert the message in our messenger RNA into an amino acid sequence that's going to be a protein or polypeptide. Okay? So transcription occurs in the nucleus. We're then going to get an RNA transcript. There's a whole process after we get that RNA transcript called RNA processing that we're going to have to perform. And then once we've done that, we're going to move the RNA out of the nucleus and into the cytoplasm, where it will bind to the ribosome and be translated into our polypeptide. Where does translation happen? Happens in the cytoplasm, at the ribosome. Now I will, for your final, something completely different on Thursday. If we get through all of this, I will show you a very, very, very funny video of this whole process happening. It was done in the 60s or 70s at Stanford. And basically it's the entire process of translation with hippies. So this should be a lot of fun if we get there. The last thing I will need to talk about that happens between transcription and translation is RNA processing, where we actually process these molecules of RNA that we make off of our DNA template. There's a very specific reason and a very important reason that we do that. Alright, so just to kind of give you an overview of how this all happens. First and foremost, differences, and then I'll let y'all go. Differences between the way these processes occur in bacteria compared to eukaryotic cells. Bacteria can perform transcription and translation at the exact same time. As the RNA is being made off of the DNA, ribosomes can be binding and making the new polypeptide because bacteria do not have a nuclear envelope. However, in eukaryotic cells, we're going to get pre-mRNA made in the nucleus. We're going to process that pre-RNA to give us our mature mRNA molecule. And only when we transfer it out of the nucleus and into the cytoplasm will we then get translation. Questions, concerns? Okay, so we'll start with the genetic code. When we come back on Thursday, we're going to get through that. Transcription, hopefully RNA processing, and translation. You guys have a wonderful day. I will see you for our final lecture on Thursday.